Martin, thank you very much for the introduction and also the opportunity to come in today and talk. And if the SIU is in Istanbul next year, I'd be delighted to meet up for a dinner. We should do something when people are in town. So for those who don't know me, and as Martin described, my laboratory and my research focus is almost exclusively focused on androgen receptor biology. And we're interested in this simply because it is the best validated pharmacological target to treat prostate cancer. But despite Nobel Prizes, 50 years of research, there's some really fundamental questions about androgen receptor biology that we just don't know. And we've been really kind of pushing the idea and the mechanism of how exactly AR drives prostate cancer biology. Now I'm going to do something a little bit against my love here, perhaps, because obviously we still are focused on the AR. But my concern is that we have hit a lot of the low-hanging fruit that's available to target the androgen receptor. We have come up with more potent anti-androgens. We've gone from steroidal to non-steroidal to the second-generation non-steroidal ones that increase their potency exponentially so. They have better pharmacokinetics. They have better blood-brain barrier penetration. We have better AR pathway inhibitors, things like abiraterone that can more effectively inhibit the production of adrenal androgens. But I'm worrying that we might have hit kind of the limit. Not to say there's not room to grow, because I think there is very clear evidence that androgen receptor is important even in the vast majority of late-stage prostate cancer. But the things that we can target, the things that we can go after, we might be getting to the point of diminishing returns. So there's a lot of work still around this. There's a lot of new inhibitors coming up, but they're moving to harder and harder targets. So you have ODM moving higher up into the androgen receptor pathway. You have these more N-terminal domains or alternative binding sites within the androgen receptor. And all of these are exponentially more challenging than a lot of the earlier work. So we're putting in a lot more work, and I'm not sure if we're going to be getting the same response out of it. So I thought it was an interesting time to take a look around for new therapeutic targets, new things that we could use to treat prostate cancer. New things that wouldn't necessarily be via the androgen receptor, but also could be profoundly important on the growth and the proliferation of it. Now, what do you look for in an inhibitor? I mean, what is the ideal target if you're going to be starting this off in a basic way? Well, you're looking for two things. One, you're looking for essentiality. You want to make sure that this target is absolutely required for the growth of the prostate cancer. But this doesn't really narrow it down that much. I mean, you can pour bleach on a cell and it will obviously kind of inhibit the proliferation, but that's not much of a drug unless you're Donald Trump. The second thing you need to have is selectivity. You need to have a target that is uniquely selective to the prostate cancer. This was shown very beautifully with these KRAS mutation inhibitors, where you could selectively inhibit only the cancer and then limit the side effects that are associated with it. So with these kind of two parameters in mind, we began kind of starting to take a look around for novel therapeutic targets that we could begin to kind of characterize and understand a little bit more. And we didn't really kind of factor in the, whether it was druggable or undruggable, kind of according to kind of classic terms. Because we figured as an academic group, we can take a swing for the fences. We can try the more challenging targets than a pharma company can in order to kind of give that proof of concept that where they can do that heavy lifting in the structure-activity relationship and all the med chem associated with it. So I'm really excited about this particular target because I think it hits a lot of these 2 points that we're talking about. There are some issues with this, obviously, but there's a lot of promise, and we're going to talk a little bit about the background today about this gene, and also we're going to talk about some of the work that we've been doing. And it's a little bit different than most of the androgen receptor because it focuses primarily on something that would be associated with developmental biology. So this is a video right here of a Drosophila embryo, and this is great to image because you've got kind of synchronized division, and also the embryos are clear. So you can see very accurately what's going on in the embryo during development. And either from the dorsal or ventral view, you can see this organizational process that occurs where the Drosophila embryo begins to kind of take a shape from kind of the original kind of cell mass into this highly organized, highly structured embryo. And this doesn't happen by accident. This is a highly coordinated, highly synchronized event that puts the right type of cells and the right type of tissue in the right place at the right time. And if any mistake happens to this, you can have profound impact on the normal development. So if you take a look at a normal Drosophila head, if there's particular mutations that occur, particularly in this group of family of genes called the HoX family members, it can really dramatically change the organization of the developmental embryo. So you can going from having a normal-looking Drosophila head on the left to having a leg coming out of the head if there's a mutation in these HoX genes. So HoX genes are homeotic genes. They're defined by having a homeobox domain, which now is quite commonly found in over 200 different proteins. And they're organized in a really well-controlled way. So you have 4 HoX family members, these 100 kilobase gene loci that are kind of found in 4 different chromosomes. So you have HoXA, HoXB, HoXC, and HoXD loci. And then there's 13 different family members in there, giving you a total of 39 HoX family members in a mammalian system. They're expressed in a highly coordinated process whereby the earlier HoX family members, like HoX 1, 2, 3, are expressed early in development, and then they go down, and then eventually kind of the later ones come up later in development. So you have HoX 11, 12, and 13 being expressed later in the development, and also kind of more found in the posterior. With regard to kind of the male reproductive system, particularly in the prostate, the HoX family members that are associated with prostate development are primarily limited to a gene that we really like called HoxB13, and that's going to be the focus of our talk today. HoxD13, HoxA13, and HoxA10. These have all been linked quite critically to the development and the formation of the prostate. If you look at where exactly HoxB13 is expressed, you can see that it is primarily found within the tail in mice and the urogenital sinus right here. If you look at the RNA expression, just with an RNA-ish, or if you generate a knock-in reporter cell line that has a LAC-Z downstream of HoxB13, you can see that this expression being found in the tail very clearly and also in the UGS as well. So as a molecular biologist, to understand function, typically what we do is just take the gene of interest, whatever we want to study, and we knock it out. We get rid of it and then kind of see a phenotype. And the last thing that you want to see as an experimentalist, the last thing that you want to see as a biologist is no phenotype. So unfortunately, when they made this HoxB13 mutation, despite this really kind of critical role of HoX family members, HoxB13 had actually very minimal effect on the normal development of a mouse. Viability was normal, fertility was normal, both for the heterozygous and homozygous HoxB13 mutation. They found 2 things that happen shouldn't be surprising given kind of where the expression occurs. The first thing was that HoxB13 mutations had slightly longer tails. I mean, the tail was maybe another kind of centimeter or so longer than a normal mouse, and with a slightly different structure in there. But in the presentation, or for the interest of the presentation today, they also observed that HoxB13 knockout affected prostate cell differentiation and secretory function primarily in the ventral prostate in mouse. So there was just a little bit different in more of a cuboidal kind of shape occurring within these HoxB13 mutations. Now, this was still a virus, something that could reproduce, but it did slightly change the HoxB13, or sorry, the activity of the prostate. But I think this is really valuable to highlight that HoxB13 is not essential in proliferation. It's not essential in differentiation. It's pretty flexible in its loss of function activity. Now, what's interesting about HoxB13 is, unlike the vast majority of HoX family members, HoxB13 remains highly expressed in adult human tissue, specifically in the prostate tissue, you can see right here, and also in the large intestine. Now, most HoX family members will decrease with time following development and will never be expressed again. In contrast, HoxB13 continues to be highly expressed in the adult human, and primarily expressed with luminal cells in the prostate, with very little expression in either basal or the stromal cells. Now, HoxB13 has been linked to prostate cancer through these very kind of elegant studies that identified a large number of germline variants that are correlated with a higher risk of prostate cancer. Most commonly, the most best characterized and studied one is this one germline variant called G84E. It's a germline variant that occurs primarily in the Nordic community at a very low rate, approximately 1% of the population. But it dramatically increases the probability and severity of prostate cancer. So it has an odds ratio of about 3.6, leading to a higher probability of prostate cancer, particularly kind of early onset prostate cancer in these cohorts. In addition, there's been some other mutations that have been found recently that have in African ancestry, primarily in the homeobox domain, But excluding that homeobox domain, the other mutations that have been linked to prostate cancer have all that same around that kind of that G8-4E kind of site. And if you look at where this occurs in the protein, here's the HoxB13 gene structure. You have two main areas, a coactivator binding domain and a homeobox domain. So this is the region that's believed to bind to DNA, and this is the region that binds to other proteins. So G8-4E is found within this mice-1 binding domain right in the middle of it, but there's others or germline variants that have been linked to higher rates of prostate cancer and severity of prostate cancer, both within this region and also in the second mice binding domain. Now, how does that kind of affect the activity then? Because if you look at the other HOX or the conservation, you can see that the homeobox domain, the region that binds onto the DNA, is highly conserved among all of the 39 homeobox or HOX family members. And there's almost no give in there. So they have a very similar sequence for all the different HOX family members. But in contrast, the coactivator binding domain, the region that actually sticks to other proteins, has a lot more variability. Hi guys, I seem to have dropped out. Where exactly did it freeze? Where exactly did it stop? Can anyone tell me approximately what slide it was on?
Yeah, it was just showing the differences between the high conservation, the DNA binding domain versus the other protein domains. That's the one. Yeah.
Thank you very much, Martin. I appreciate that. So as I was saying, the homeobox domains are highly conserved at the part that sticks to the DNA. The motif is commonly found all throughout the genome, and there's not a lot of give within that. And in contrast, there's a high degree of heterogeneity within the coactivator binding domain. And the reason why this occurs is that the HOX family members, the homeobox domain family members, their activity is altered by context-dependent protein-protein interactions. What exactly is bound to the HOX family member will impact where it binds and also kind of what it will affect in the gene transcription. And this can be seen very clearly in the case of HoxB13. Sorry, guys, I'm terribly sorry. I think I've got it sorted out now.
Okay.
Okay, thank you. Okay, can people hear me okay?
Yes.
Okay. So as I was saying, HoxB13 interactome dramatically changes during development and progression of prostate cancer, where in the normal prostate cancer or normal prostate tissue, HoxB13 is highly bound and interacts quite closely with these mice-1 and mice-2 family members. And when it does this, when it's interacting with mice-1 and mice-2, HoxB13 is primarily a tumor suppressor. It prevents the development and the progression of prostate cancer. But when mice-1 is downregulated, HoxB13 then changes its partner and begins to interact with the androgen receptor. And when that happens, HoxB13 activity dramatically changes and actually kind of pushes to the growth and the proliferation, where it changes where the AR can bind. You see kind of an expansion of the AR systrome, where the DNA binding sites to be far more neoplastic-like than like a normal prostate cancer or normal prostatic tissue. In addition, it tends to interact or change the interaction pattern of the androgen receptor variant, these constitutively active splice variants as well. In addition, there's been some recent evidence to suggest that the AR-mediated transcription can be dramatically affected by HoxB13, both increasing and decreasing androgen receptor regulated genes in a highly locus-dependent manner. In addition, this change can then perhaps change or affect the growth and the proliferation of the disease, though this is a bit of a controversial thing, mostly because it's not really well understood if HoxB13 is itself regulated by the androgen receptor. Because I think there's a narrative associated with the AR, because the AR is so important in prostate cancer biology. It is such a focus of research that we tend to think of everything through an AR prism. And in the case of HoxB13, there's been conflicting evidence of whether the HoxB13 itself is regulated by the androgen receptor, with some studies saying that it's a FOXA1 mediated event, while others show that castration tend to decrease or alter HoxB13 expression. But I think this is too narrow a prism, because HoxB13 is one of those genes that is so critical. It is so important in the kind of normal lifespan of prostate cancer, that it seems to be that it can do far more than just work with the AR. It's more than just kind of driving the AR. And giving evidence to this potentially kind of important functionality is that there have been studies that have demonstrated quite clearly that HoxB13 can impact prostate cancer growth, though this is somewhat controversial with some conflicting studies as well. So with this information in hand, with these results available to us, we then kind of start to dig into whether this could be something interesting, whether this could be an interesting pharmacological target, because we ideally would be looking for something outside of androgen receptor activity. We love the AR. We're happy to kind of work on understanding how the AR goes, but we're looking for therapeutic targets that are not androgen receptor driven, that have kind of an independent functionality. We also then start to take a look at whether the HoxB13 was essential for the growth and the proliferation of prostate cancer. So the essentiality of it, looking at both in vitro and in vivo. And finally, because we are basic biologists, looking at the mechanism of this. How does HoxB13 affect prostate cancer activity? So with these three specific aims, we went through each of them before kind of committing to HoxB13 as a target, because we want to be sure about this. We want to do that background to ensure that it truly is a good target, that it has that selectivity, that specificity that we're looking for. So with regard to the relationship with the androgen, the first thing that we did, because I'm a scientific vulture. If there's any data that's available out there, I'm going to kind of grab it and analyze it for our own particular question of interest. Is that we took advantage of a recently published data set of single cell RNA sequencing from a primary prostate cancer cohort. And when we look at this, when we kind of annotate each cell type within this, we can find very clearly that HoxB13 is expressed primarily in androgen positive luminal epithelial cells. This matches what we see in immunohistochemistry, with AR being found in stromal and endothelial cells, but this isn't the case for HoxB13. It's highly kind of specific to these luminal epithelial cells. When we went on to take a look at the in-house patient cohorts, these tissue microarrays that the Vancouver Prostate Center had set up over the years, we found that HoxB13 expression does not always correlate with AR status. So within primary prostate cancer, castrate-resistant prostate cancer, treatment-induced or neuroendocrine prostate cancer, there's very high levels of HoxB13 expression in those cells that are AR positive, which is not surprising, kind of given the well-known kind of relationship in HoxB13 and cell identity. But what was interesting is if you take a look at the treatment-induced NEPC population that is AR negative, so these have no AR present in this small cohort, you still see relatively high levels of HoxB13 in a percentage of this population. It's not all of them, but it's also not nothing either. So in about 40% of these patients, we see some level of HoxB13, either medium or high. So this does suggest a little bit that HoxB13 is not necessarily regulated by androgen receptor. And to give it a little bit more kind of credence to this, we had the wonderful kind of cohort that Martin had put together over the years in this neoadjuvant setting, where we have a patient cohort of high-risk prostate cancer patients, either who have received ADT or not before kind of doing the radical prostatectomy and collecting the biopsy. And in this neoadjuvant cohort, we see that the ADT treatment does not actually change the expression of HoxB13. Once again, highlighting that HoxB13 is not likely to be androgen regulated. It is independent of AR, at least in its gene regulation. Now, going on to take a look at the essentiality, we took advantage of some of the more modern kind of gene modulation approaches, and we did initially just a CRISPR dropout of a panel of prostate cancer cell lines versus kind of normal or normal-ish non-prostatic tissue. So when we do this, and we do kind of three guide RNAs, three individual guide RNAs targeting three individual parts of the gene loci, we can see very clearly that prostate cancer cells are highly dependent on HoxB13. So if we knock out HoxB13 in every one of the prostate cancer cell lines we've tested, excluding DU145s, which do not express HoxB13, you can see that they're essential for the growth and the proliferation of the prostate cancer models. So even AR negative cell lines like PC3s, when you get rid of HoxB13, cell growth goes down to almost nothing. And this is very quick. This occurs within a 3-day to 4-day period, which is quite distinct from the androgen receptor. In contrast, non-prostatic tissues like 293Ts or HEP3Bs are not affected by knockdown of HoxB13. These results were then confirmed with an orthogonal methodology. So we use an RNAi, shRNA or shRNA system to kind of show that knockdown of HoxB13 was required for prostate cancer growth and proliferation. And I just want to highlight once again that this is not likely due to the AR, simply because the kinetics of it just don't work out. If you knock out the androgen receptor, you don't really see cell death. You don't see kind of apoptosis. You see a decrease in proliferation over a long period of time before eventually the cells just kind of stop growing. They die at the very end of it. But this takes, 10 to 12 days before you see a phenotype. In the case of HoxB13, this happens in a 3 to 4 day period. Now, it doesn't definitively say it's not through AR, but it does kind of suggest that it is kind of through a higher upstream mechanism. Now, what's really interesting is that nowadays, there's these great resources available, including genome-wide CRISPR screens. And this is done by the Broad Institute, where they literally just do genome-wide CRISPR screens on every single cell line they can get their hands on. This doesn't matter what the model is, doesn't matter what the question is. They will knock out every gene and then kind of see what kind of effect it has on growth and proliferation. So they've done this for almost 1,000 cell lines by this point. And when you actually take a look at the essentiality of HoxB13, so what's required for growth and proliferation, it's primarily and almost exclusively prostate cancer cell lines that require HoxB13. In the vast majority of cancer cells or cell lines, if you get rid of HoxB13, it has no effect at all on their growth, kind of giving credence to that selectivity that we talked about before. Now, we do see some cell lines that don't have HoxB13 dependency, but these are also cell lines that are not commonly used as an epithelial model. They're either kind of normal or normal-ish, or they have kind of no AR being found as well. When we characterize the mechanism, we see that using an inducible shRNA method, the loss of HoxB13 induces this very acute apoptosis that we can observe, primarily in the prostate cancer models. But what was really exciting to us is when we started to take a look at the essentiality within an in vivo model. We did this two different ways. We looked, first of all, whether HoxB13 was required for tumor engraftment. So we took our prostate cancer model and we either gave it a non-targeting guide RNA or a HoxB13 targeting guide RNA, and then injected it into the mouse and tried to take a look at what would occur with tumor engraftment. We could see very clearly that the non-targeting guide RNA had normal growth, proliferation. The xenograft led to a visible tumor that was palpable and then eventually kind of would grow out of control. In contrast, though, the HoxB13 knockout had almost no tumor uptake at all. We could see barely anything within the xenograft site or injection site. And this was true whether it was an AR positive model like an LNCAP or an AR negative model like PC3s, where HoxB13 was required for these xenograft engraftment models. Now, that's great, but that doesn't really tell you whether it's suitable in a growing tumor to target HoxB13. It doesn't give you that confidence that you're looking for. So to test for this, we took a slightly different approach where we let the tumor grow. So there was a palpable tumor, that there was something that we could clearly feel, and then we gave it a DOX-inducible shRNA. So we would either knock the gene down or give it a non-targeting control once the tumor is already formed. So in this experiment, we could see that both the non-targeting or the HoxB13 targeting had a relatively normal rate of growth within the initial engraftment before we gave doxycycline. In fact, the HoxB13 knockout or the HoxB13 shRNA expressing line had higher growth rates until we gave doxycycline. And when we gave this, the shRNA was produced and then kind of knocked down either the non-targeting or the HoxB13, leading to a decrease in growth, specifically in the HoxB13, but not the non-targeting, highlighting that HoxB13 is essential for PCA growth in vivo in these xenograft models. One thing that we also found that was quite interesting is that if we take HoxB13 and we put it in a cell line that doesn't express HoxB13, specifically the DU145, we could observe a increase in growth either with an MTS assay or other orthogonal methodologies. And this was actually a really nice tool because then it began to allow us to tease apart how HoxB13 activity contributed to proliferation, where we could make a series of mutations in wild types and then kind of map exactly what was required for HoxB13 mediated proliferation. And we saw very clearly that the mice interacting domain, either mice interacting domain 1 or 2, were required for the increased proliferation. And actually loss of the homeobox domain did not have an effect, suggesting that it's not mediated through this HoxB13 DNA binding or HoxB13 transcription factor biology. But then digging into the mechanism, we got into kind of trying to figure out exactly why HoxB13 was so critical in these models. We felt really confident about our results. We'd proved it, several different ways, showing that HoxB13 was essential in these prostate cancer models. But the question is then why? And specifically, why is it so important in these AR-dependent and AR-independent models? Because HoxB13 activity in the mechanism has almost always been focused on AR biology. And we had this really interesting finding suggesting that HoxB13 activity was not limited only to AR and that it could work on other transcription factors. So to figure out how this was working, we then first of all, we just looked at where the DNA binding occurred. So we took our HoxB13 cell lines, the LNCap cell lines, which is AR positive, and then PC3, which is AR negative, and we did a ChIP-seq. We tried to find out where exactly HoxB13 was binding throughout the genome. And we could see very clearly that they both required HoxB13, they both needed HoxB13 to grow, but their activity was totally different. The binding sites of LNCap were not the same binding sites as PC3 cells. There was very little overlap between the two of them. And we started to dig into exactly why that may be. And we could see very clearly that the HoxB13 binding sites overlap with really distinctive transcription factors, where the LNCap, unsurprisingly, had a high overlap with androgen receptor motifs, FOXA1 motifs, GATA2, while in contrast, the PC3 cells were more likely to interact with AP1 transcription factors. So you had this change in behavior for HoxB13 where it had been interacting with the androgen receptor, but had transitioned to begin to work with other AP1 like transcription factors as well. To kind of confirm this as well, we also looked at published ChIP-seq. So this is every piece of published ChIP-seq available. And we could see that the motifs of what exactly was interacting with were changing. You were seeing different transcription factors overlapping with the AR and the PC3, kind of giving further confidence to that change in selectivity or transcription factor interactions. But the only thing that was kind of common in the two of them was this thing called SMARCA4. And SMARCA4 is a member of a group of proteins called the SWI/SNF family members. And these are chromatin modifying proteins. This is a group of proteins that come together and working as a complex allow DNA to be opened, giving space for a transcription factor to bind in there. And this was the one thing that came up as a common hit in both of these things. But that doesn't really kind of nail down exactly how HoxB13 affects the activity in PCA growth. I mean, we could see this change in binding, we could see this change in overlap with different transcription factors, but given that they were equally essential in these two different models, there had to be some sort of common pathway whereby HoxB13 would bind these transcription factors, but then would activate or modulate behavior in some consistent way in both of these two distinct systems. So to look at this, to identify what the interacting proteins were, we did something called rapid immunoprecipitation mass spectrometry, which is a system whereby you take your cells, you cross-link them, then you pull out your protein of interest, which in this case is HoxB13. We isolate our digested peptides, and then we figure out what exactly these peptides are through mass spectrometry. And this will identify all of the interacting proteins of HoxB13 on chromatin. This was done in 3 different models. 2 of them were AR-dependent, an LNCap and 22RV1s. And then finally, we had that PC3 AR-independent model. When we look at them, we could identify approximately 70 to 100 different proteins that interacted with HoxB13 in each of these different models. And what came up as common interactions in these was very interesting. So within the AR-dependent interactome, so this is either in the LNCap or 22RV1 system, HoxB13 was primarily interacting with AR, NKX3.1, FOXA1, topoisomerase 1, topoisomerase 2, things that we know work with the androgen receptor to drive transcription. What was interesting is that if you look at the common interactome, which were found in both the AR-dependent and independent models, HoxB13 was also interacting with a protein which is called SMARCD2, which is a member of that same SWI/SNF family member that we talked about. And if you look at kind of where it was within these hits of PC3 versus LNCap, you can see that SMARCD2 was a common hit in both of these two models, as well as additional members of the Swiss or SWI/SNF family member or complex. So from this interactome study, we obviously had to validate it. We had to show whether this was real or not, or just an artifact of these large-scale kind of rhyme-like experiments. So to do this, we did proximity ligation assay, which is a very fancy way, or a very easy way actually, of looking at protein-protein interactions by counting the number of dots that are produced through rolling amplification. So if you see a red dot, you're seeing interactions. If you don't see a red dot, you're not seeing an interaction. So we did this in several different models, including some that do not have HoxB13 in that DU145 cell line, and then the PC3, 22RV1, and LNCAP, which do have that dependency on HoxB13. And we could see very clearly that there was this interaction between HoxB13 and SMARCD2 in the HoxB13 expressing models. And then if we took the DU145 cell lines and we overexpressed the HoxB13, where we see this increase in growth, we could observe a number of interactions between HoxB13 and SMARCD2, kind of giving further confidence to this. Now, nobody likes one assay, so to prove that this was real, we also took another step back and did an immunoprecipitation and showed that you could also have this SMARCD2 HoxB13 interaction as well, and that this interaction primarily happened through the mid-1 and mid-2 domain, because when we disrupt these genes or these domains genetically, we could see that drop in interactions. Now, that's great for a cell line. To look at kind of in a clinical cohort, we then tried to correlate the expression of HoxB13 and SMARCD2, and we see a very nice, clear correlation between these two genes in primary prostate cancer. In addition, we did the PLA within prostatic and non-prostatic tissue, and we could observe, once again, a HoxB13 SMARCD2 interaction specifically within prostate cancer, but not within the non-prostatic tissue. What was interesting is that we didn't see a huge change in SMARCD2 HoxB13 interaction during prostate cancer progression. Between normal prostate and CRPC, or even treatment-induced AR-positive NEP-C, there was still a high level of HoxB13 SMARCD2 interactions, suggesting that may be a common mechanism, regardless of whatever transcription factor is interacting with HoxB13. Now, to understand the functional impact of this interaction, to understand how HoxB13 interacting with SMARCD2 can change the behavior, we did something called an assay for transposed accessible chromatin using sequencing, which is a bit of a mouthful, I will admit. But what ATAC-seq does is essentially it tells whether the genome is open or is closed, whether you have euchromatin or heterochromatin. And an ATAC-seq is a very easy way to look at the accessibility within the genome, to see how open that particular region is. Now, we did this with the simple concept that the SWI/SNF family members are known to open up chromatin. So theoretically, if HoxB13 is interacting with this SMARCD2 and the SWI/SNF family member, we should see increased chromatin accessibility at these regions. So what we did is we took DU145 cell lines that do not express HoxB13, and we overexpressed either something that doesn't have any activity, this GFP protein, or HoxB13, and then we took a look at the change in accessibility across the genome. And what was really interesting is that we didn't see new sites being formed. Overexpressing HoxB13 did not change the accessibility of new sites. You didn't have gained sites. You didn't have chromatin opening up in regions that were heterochromatin before. But what you did observe is that in the HoxB13 overexpressing cell lines, there was a lot more accessibility broadly across almost all the sites. And when we start to take a look at this distribution, you can see the change in the HoxB13 versus the GFP. There's a shift in exactly kind of the accessibility across the genome. And when we start kind of separating that to the top 5% versus the bottom 95%, we can see that the top 5% of these sites, the sites that are the most open following HoxB13 overexpression, tended to enrich for motifs associated with HoxB13. So they had DNA sequences that you typically would associate with HoxB13 or AR family members. We then overlaid that with HoxB13 ChIP-seq from our LNCAP model that we previously had done. And we showed that the 5%, these highly expressed, accessible regions or more increased accessible regions were very likely to interact with HoxB13 ChIP-seq peaks as compared to random or the CRE sites. This was then further carried on when we start to take a look in a primary and CRPC cohort where there is ATAC-seq available to them. And in this patient population as well, we observed that the HoxB13 enriched sites were much more likely to interact with accessible regions in primary prostate cancer or CRPC as compared to random or the EGFP enriched ones, suggesting that this HoxB13 overexpression does correlate with increased accessibility. To finally nail this down, we then took that same experimental system where we overexpress HoxB13 and then knock down SMARCD2 using an siRNA and did that same attack-seq experiment. And when we do this, we see a clear decrease in the HoxB13 accessibility, or the HoxB13 enriched accessibility, supporting our mechanism whereby HoxB13 SMARCD2 kind of changes chromatin accessibility. So to kind of confirm this a little bit more, we then did a ChIP-seq experiment of SMARCD2. So this will tell us the binding sites of SMARCD2 across the genome. We already have the binding sites from HoxB13, and then we can kind of compare where they fall. And we can observe very clearly that SMARCD2 largely overlapped very well with HoxB13 peaks. So the peaks that we observed with HoxB13 were quite likely to interact with SMARCD2, though there were regions that did not strongly overlap with SMARCD2, but they did have strong HoxB13 peaks. And there was also regions that had high levels of SMARCD2 peaks, but almost no HoxB13. And when we took a look at kind of the activity of these sites, what these sites were doing, focused on, that measures the enhancer activity. So whether something can kind of modulate gene transcription, we observed that the SMARCD2 HoxB13 overlapping sites had high levels of enhancer activity, while in contrast, the HoxB13 alone sites had very low levels of enhancer activity, suggesting that HoxB13 SMARCD2 interactions in this mechanism were increasing the accessibility and then kind of modulating the enhancer activity. And that this interaction is likely stabilized by transcription factor binding, because when you take a look at these same kind of ChIP-seq tracks and you look at where the androgen receptor binds, you can see that HoxB13, SMARCD2, the common sites where both of them are present is much more likely to have an androgen receptor present than the HoxB13 alone sites. And that interaction between HoxB13 and SMARCD2 can actually be stabilized by the presence of androgen receptor. So if you take a model and you look at the HoxB13 SMARCD2 interactions, either with androgen receptor or without androgen receptor, in the case of the ethanol and DHT, you can see that there's an increase in HoxB13 SMARCD2 interactions mediated through this androgen receptor interaction. So our working model so far is that HoxB13 will interact with a particular transcription factor, whether it be androgen receptor, MICE1, or AP1. And then through this interaction, it will stabilize the SWI/SNF complex through SMARCD2. This then kind of alters chromatin accessibility and then increases transcription. And in the case of androgen receptor or AP1, it increases proliferation. So in addition to this kind of more mechanistic and validation studies, we were very encouraged by our in vitro and our in vivo data that we've pushed this forward as a potential drug target. And we've done a lot of biochemistry developing novel assays that can quantify HoxB13 activity or HoxB13 DNA binding. And we're starting to begin to do more structure-guided drug development, as well as kind of more novel, non-conventional small molecule approaches to identify HoxB13 inhibitors. So in conclusion, HoxB13 is highly expressed in both prostate cancer, and it's not an androgen-regulated gene. HoxB13 seems to be essential in prostate cancer growth. In almost all the models we've looked at, HoxB13 was required for growth and proliferation. And finally, that HoxB13, the mechanism of action is likely via this SWI/SNF interaction that then changes the chromatin accessibility within the genome. So with that, I'd like to thank my funding sources and also the people who did this work.